Good afternoon and welcome to the launch webinar for the BBSRC Follow-on Fund. My name is James Phillips and I'm pleased to be speaking to you today at the very start of our first round for FOF in 2024. The Follow-on Fund, or FOF, is one of BBSRC's flagship programs. We're looking forward to celebrating 20 years of investment this year and a fantastic record of researchers accessing funds to progress their ideas towards a huge range of products, services and processes, generating both economic and societal impact. Today's webinar will run from 1 until 2. We'll be taking you through the support that's available, and this includes explaining the requirements to apply for the funding and what's involved in actually making that application. We also have presentations from guest speakers to showcase examples of successful follow-on fund awards. At the very end of the session, there will also be a live Q&A session for you to send us your questions directly. Throughout the webinar, we invite you to send us your questions using the Q&A function on Zoom. Please press the Q&A button on the toolbar at the bottom of the window to add your queries. We'll try and answer everything submitted today, but if we're not sure, then we will work to send you a written reply after the webinar finishes. We do plan to keep a record of questions submitted to help us write an FAQ document for the website afterwards. I'm joined on the webinar today by colleagues who lead FOF. I'm very pleased to introduce the fund's Strategic Portfolio Manager, who is Faye Shannon, and the Senior Lead for the delivery of the fund, who is Ellie Tayaki. Between us, we hope to be able to answer all of your questions today. However, there are two things that we would ask you to note. The first is that the follow-on fund is delivered through the funding service. If your question is specifically about how to use TFS, then we would encourage you to please email the TFS support desk directly. Similarly, if your question is very specific to your project, so for example, if you want to check whether your idea is within the remit of BBSRC, then we would encourage you to please send us an email at our team mailbox, which is FOF, that's FOF, at bbsrc.ukri.org. We will try to answer all questions today, but when it comes to project-specific queries like scope, remit, and arrangements for intellectual property, it's much better to use email so that there is a clear written record of the advice that we've issued. I mentioned earlier that FOF is a flagship program for BBSRC. As an investor in research and innovation, we have an ambition to advance the frontiers of biology, and the follow-on fund is a really integral part of how we deliver that vision. It provides dedicated funding for BBSRC award holders to progress their ideas towards products, services and processes with the potential to deliver impact. The fund supports early stage projects that de-risk innovative ideas and later stage projects with significant technical milestones. Successful applications will be within the remit of BBSRC and will demonstrate scientific and technical merit, economic and societal impact, and a strong potential for added value. They will also have a clear approach to managing any IP in line with the guidance on the additional information section of the Opportunity webpage. This slide summarises the support that's available. The follow-on fund will make grant awards that allow standard costs from a total fund of £2.25 million in this round. Awards may range from £100,000 to £800,000 in value. Please note that this is the 80% FEC figure, and it represents the value of the award from BBSRC to the research organisation. We estimate that the earliest start date is the 1st of October, and awards may run from this date for up to 24 months. This time round, the closing date is the 27th of March at 2024 at 4 p.m. Please do remember to submit your application to your internal approvers well before that date. Late applications unfortunately cannot be accepted, but we are planning a second round of FOF this year, which should launch in the summer. So I'm now going to switch topics to talk about the strategic context of the follow-on fund to help explain what the fund is and exactly what we're looking for. BBSRC is one of the 9 bodies that make up UKRI, which is the UK's research and innovation agency. UKRI was formed in response to a variety of needs, but one of those goals was to ensure that public investment in research progressed towards innovation and impact. BBSRC is the research council that focuses upon biotechnology and the biosciences. We have a good track record of providing dedicated knowledge exchange and commercialisation activities to unlock the potential of biology. This is reflected in UKRI's vision for research commercialisation, which emphasises the importance of supporting this activity in the research base. UKRI's vision includes working to influence and incentivise collaboration and openness, advocating for research commercialisation, which includes generating evidence, and maximising support through dedicated funding routes such as the follow-on fund. This focus is also reflected in BBSRC's strategic delivery plan, which describes a vision to support knowledge exchange and commercialisation activities that will enable a vibrant bioscience ecosystem that connects world-class researchers with leading innovators to advance national priorities and access shared opportunities. This recognises the complexity of the biosciences and the need for flexible and open support mechanisms. This priority is delivered within BBSRC by the Knowledge Exchange and Commercialisation team, which has an overall mission to enable a more connected innovation system that delivers for bioscience across the UK. The team manages strategy and policy relating to KC across research, innovation, infrastructure and skills, and this includes direct support for translation, capability, venture and enterprise. The follow-on fund is one of the translational programs managed by the team and is central to delivering UKRI and BBSRC's strategic goals for KC. We're seeking proposals that fit within this strategic ambition, and particularly those with the potential to progress bioscience research towards a product, service or process, and that includes outputs such as spin-outs or licensable IP. All of which reflects our vision as a funder to enable researchers to progress their ideas from discovery towards achieving impact as part of supporting a successful bioscience community. So that concludes the introductory session. We're now going to hear from researchers who were successful in applying for funding from FOF and how this has enabled their ideas to progress. Thank you.
Hello everybody, my name is Matthew Paul from Rotherton Research, and I'm going to be telling you a history of our research on the trihalo-6-phosphate T6P sugar signalling pathway and how this has gone from discovery to translation and how the follow-on fund helped at a critical stage. The translation aspect has been through a superb collaboration with Professor Ben Davis at Oxford University, and from 2014 Cara Griffiths as the main researcher, who is now the Chief Technical Officer of SugarOx, our spin-out company. So going back to the late 1990s, the trihalose pathway was thought of as a specialized pathway in extremophiles, such as resurrection species, where trihalose accumulation enabled plants, such as this Selaginella species, to survive and recover from almost complete desiccation. At this time, it was thought that this property could be engineered into crops to make them drought resilient. What genetic engineering of the pathway into plants did do, however, was to uncover a central mechanism that determines the use of sucrose and regulates sucrose homeostasis, or the plant equivalent of blood sugar. And as we now know, T6P integrates metabolism with development and controls important crop traits such as reproductive development, and promotes anabolic metabolism such as the synthesis of starch, a yield component of many crops including cereals. This slide illustrates the journey and the important part funding from BBSRC has played. The discovery of the indispensable nature of T6P was discovered in 2003, and its mode of action through the protein kinase SNRT1, a universal energy regulator in plants, we published in 2009. Then in 2006, funded by the Selective Chemical Intervention in Biological Systems initiative from BBSRC, we set on a path towards the spin-out company with Ben Davis, with the idea behind this was to design plant permeable T6P sprays as an alternative to genetic modification, i.e. chemical intervention, which could be more straightforward and flexible and potentially be used on many crops. The chemistry worked well, and as I shall show in a moment, led to a patent application as a means to increase starch content and yield of crops in 2011. Then in 2016 we published the work in Nature, which led to the formation of the spin-out company thanks to follow-on funding that has supported the translation of this work. As you can see, there are other grants that have been funded with other partners such as Matthew Reynolds at CIMMYT, Mexico, that have enabled us to build up research momentum in this whole area. Going back to 2006 and the development of the technology, essentially it centred on producing trehalose 6-phosphate precursors that could be taken up into plants and released upon exposure to UV light or sunlight. T6P in its native form is not taken up by plants. So this had to be dealt with by attaching a light-sensitive group to T6P, which then could be cleaved off once the compound was in the plant. Proof of concept that this could work was shown in Arabidopsis, where it was possible to increase the starch content of Arabidopsis plants. If we could increase starch content of wheat, this would be potentially a way to increase yields of crops, such as wheat and other cereals. And in fact, that's what's happened. Spraying T6P compounds at the start of the grain filling period at 10 days after anthesis resulted in more starch in larger grain, increasing yields by up to 18% in glasshouse conditions. Our follow-on fund then enabled us to take this to the next stage, which was to improve the synthesis pathway by reducing the number of steps using trehalose as a starting material to make it economically viable for farmers to spray this onto their crops. And secondly, we had to show that the technology worked in field conditions. Many technologies that work in the laboratory do not translate to the field, and this includes many technologies, a whole spectrum of technologies, including medical ones. So this slide shows the synthesis of large amounts of chemical product through an improved synthesis pathway that we can now produce the compound economically. Field trialling was conducted in Argentina using 7 meters long plots over 4 seasons, trialling 3 Argentinian elite varieties, Cibo, Inter and Cita, as you can see here. By chance, we had 2 wet years and 2 dry years. The wet years gave yields of between 5 and 6 tonnes per hectare, whereas the dry years gave yields of between 2.5 and 3.5 tonnes per hectare. In each of the 3 years of trialling, we had statistically significant yield increases, as you can see by the p-values against each year there towards the right of the slide. Combined analysis over all 4 years, also using a mixed model, also showed that these results were highly significant. Even in 2021, which is the graph on the far left of the slide, the bottom left of the slide, when, because of the pandemic, our compound arrived late in Argentina, meaning that application was not until 16 days after anthesis instead of 10 days after anthesis, we still saw significantly increased wheat yields. So there is a window of between 10 and 16 days after anthesis for our spraying to achieve a yield increase, making it easier for farmers to operate. Argentina is a good environment for testing because it is representative of global wheat production, with yields that are close to global average and with variable rainfall, which is typical of much of global agriculture. Yields have been increased without extra nitrogen, and grain protein was not diluted out in the larger grain. So yields are increased per unit of nitrogen and per unit of water. And you can see that in the wet years we had 12.7 on average more yield, and in the dry years we had 9.2% on average more yield. It appears that the technology works by upregulating the whole starch pathway from sucrose import into the grain all the way up to amylose and amylopectin synthesis, enhancing zinc strength, which results in an increase in the rate of photosynthesis as well, hence coordination of source and sink. We also find improved vascular connections in the cross-sections of the treated grain, shown here in the middle of the slide, enabling delivery of sucrose to the filling grain. We've also tested barley and sorghum and have found they respond too. And here is the result with sorghum, showing quite a dramatic increase in the yield of the sprayed plants on the left of the screen. So this central mechanism in the middle of the slide that came out of responsive mode funding and then with follow-on funding increases wheat yields in field conditions when we modify it with our chemical spray. We've also been able to show with collaborators in Ghent using our light activated compounds that T6P regulates lateral root growth, and this is shown in the top right hand corner of the slide. We are also conducting parallel genetic research to dissect out mechanisms through which the T6P pathway regulates reproductive development, shown on the left of the slide, performing gene editing and looking at natural genetic variation in trehalose phosphate synthase and trehalose phosphate phosphatase genes. The whole journey over 25 years certainly develops character, and you need to be patient and resilient and stick with things. Trust your instinct, focus on your goal, and bring good people with you, and collaborate internally, externally, and internationally with people that have complementary and interdisciplinary skills. In our case, this was the chemistry-biology interaction. Obviously, we've also worked with scientists that really understand how to do field trials as well. And we've diversified our funding streams, and we've patented and published the work. All these points are really, really important to ensuring that you can reach your goal. I've mentioned several people along the way. Many more people have been involved over the years than I could probably fit onto the slide. I'm very thankful to all of them. It wouldn't have happened without them. This slide shows the people who have been most recently involved, particularly with the yield effects in wheat and also the lateral root work. And thank you to BBSRC.
Good afternoon. My name is John Walsh, and the title of my talk is Virus-Resistant Crops. I'm based at the School of Life Sciences at the University of Warwick. So I've been asked to talk you through our initial research and then the follow-on project and finally the exploitation phase of our research. So first of all then, the problem. So this is turnip yellows virus in the crop oilseed rape, and the solution. So the solution as we see it is natural plant resistance, and then I'll take you through the journey. So this involved BBSRC funding in the form of a Crop Improvement Research Club grant, followed by the follow-on fund grant. And finally, the destination. So the destination is the exploitation of natural plant resistance by the plant breeding industry so that farmers can utilize it to improve yields and also reduce their reliance on pesticides. So back to the problem. So turnip yellows virus in oilseed rape. So the virus has very high incidences in rape fields in the UK and also in the rest of Europe, when we've recorded 100% incidences on a number of occasions. So HDB have put the annual losses at around 9%, representing £60 million worth of losses to farmers in the UK. So the virus is transmitted from plant to plant by greenfly, or aphids if you prefer, particularly the peach potato aphid, Mysis persicus. And once the aphids acquire the virus, they have it for life, so they can go through a winter with the virus and then come out in the spring and still be able to transmit the virus. The ban on neonicotinoid insecticides is considered to have exacerbated virus problems in crops, including turnip yellows virus in oilseed rape. So in terms of the solution, I said that our idea of the solution is natural plant resistance. So a source of natural resistance was identified some years ago and has now been incorporated into oilseed rape. The yield benefit of this resistance is so good that it's now been incorporated into most cultivated commercial varieties in Europe. But of course, this creates a problem. That's the one of extreme selection pressure for resistant breaking strains of the virus. So new sources of resistance were needed to reduce this selection pressure for resistance breaking strains of the virus, and also to manage the current resistance and also new sources of resistance. So in terms of this solution of natural plant resistance, in evolutionary terms, oilseed rape, also known as Brassica napus, is a new plant species which has resulted in restricted diversity. Screens of oilseed rape germplasm have revealed very little resistance to the virus. So you can see here Brassica rapa, a diploid species with the A genome, and Brassica oleracea, a C genome species, have hybridized, or is a natural hybridization, resulting in the origin of oilseed rape. So you can see oilseed rape is an amphidiploid, and it's got the AA genome from rapa and the CC genome from oleracea. So the outcome of this is the thought that oilseed rape arose from a very limited number of hybridizations between rapa and oleracea. So this is a genetic bottleneck, further exacerbating the restricted diversity in oilseed rape. So in terms of the journey to try and generate virus resistance, we'd identified natural source of resistance into Brassica oleracea, and funding was needed to exploit this resistance with a view to incorporate it into our commercial oilseed rape crop varieties. The initiation of a BBSRC research initiative called Crop Improvement Research Club, or CIRC for short, was driven by Dan Godfrey at BBSRC. And the initiative was subsequently managed by James Phillips, Jane Brookman, and also the late Simon Bright. So we applied for funding from the CIRC initiative, and we were successful. The initiative worked very well in our eyes, and representatives of the seed breeding companies that had contributed financially to the club attended regular meetings, along with colleagues from other organisations, including universities and research institutes. You can see here a list of the commercial collaborators. So participation was very positive, supportive, and also a very beneficial experience for us. So this BBSRC CIRC grant allowed us to progress our research on turnip yellows virus resistance in oilseed rape. I mentioned that we'd identified resistance in Brassica oleracea prior to applying for the funding. So we then went on with the funding to identify sources of resistance to the virus in the other oilseed rape progenitor, Brassica rapa. So we then developed mapping populations for the turnip mosaic virus resistance in Brassica rapa and also in Brassica oleracea, and we identified quantitative trait loci, or rather QTLs, for resistance in both species. So that's the mapping of the resistance. We were then able to take the resistant rapa line, the resistant oleracea line, and use them to, in terms of crossing, to resynthesize the amphidiploid AACC plants, and this is of course the oilseed rape genotype. This isn't a particularly easy process. You can see here it involved microspore culture, etc. And we also, as part of the project, studied the genetic variation of turnip yellows virus in Europe and studied the stability of the resistance to different TUIV genotypes. In terms of the outcome, a PhD student who also worked on the project now has a permanent position with Syngenta. So the next steps of the journey. Having identified new sources of resistance in Brassica oleracea and Brassica rapa, mapped these resistances and produced the resynthesized AACC plants, the next steps were to map the QTLs, fix the resistances, and hopefully hand over to commercial plant breeders to exploit the resistance. So we put together a project team at Warwick with a number of commercial plant breeding partners, including Syngenta, Limagram, Elsams, and LS Plant Breeding. And the idea, of course, was to deliver these amphidiploid AACC plant lines with the resistance fixed and molecular markers for the QTLs that would accelerate the integration of the resistance into commercial oilseed rape varieties. You see the partners here. So we applied for a BBSRC follow-on fund, and fortunately we were successful, meaning we could continue with our research to exploit the resistance. So the project involved 3 quite complex work packages. You can see an example here, but to simplify things, I put all the work packages into this one slide. So the project, the follow-on project, was a truly integrated project where all partners had different roles that were independent. So starting then with the virus resistance that we had found in Oleracea and Rapa and the resynthesized Napus line that we produced, we passed seed of the resynthesized Napus line to LS plant breeders, and they used this to resynthesize and genotype DH lines of Brassica napus that was segregating for the resistance to turnip yellows virus. We also passed seed of the diploid species, Brassica oleracea and Brassica rapa, that was segregating for resistance to turnip yellows virus to Elsams, and they produced further lines that were segregating. And some of the lines were also DH, doubled haploids. So we phenotyped and genotyped some of these segregating populations, and the DNA from the plants that we had phenotyped and genotyped was sent, at least the Oleracea DNA from the Oleracea plant was sent to Syngenta, and they did further genotyping of the segregating Oleracea populations. We also passed DNA from the plants that we'd phenotyped to Limagran. In this case, it was the Brassica rapa plants, and they genotyped these plants. So in the end, we had fixed resistance to turnip yellows virus in the AACC oocyte rape genotype, and Syngenta and Limagran shared all their genotyping information with all the other partners, which has allowed them all to develop their own molecular markers, which can now be used to integrate the resistance, or to speed up the integration of the resistance into commercial oocyte rape varieties. So seeds of this Napus material is already with the seed companies, and hopefully they are making a start in moving the resistance to turnip yellows virus into commercial plant varieties. So to acknowledge the other people involved in this research project in terms of the CERC funding, the co-investigators were Graham Teakle and Guy Barker, also at Warwick, PhD student Max Newbert, who I mentioned before, and the postdoc was Adam Baker. And in terms of the follow-on fund team, again, Guy and Graham were the co-investigators. The postdocs were Dieter Hackenberg and latterly Kyle MacLeod. And of course, we have to thank the commercial partners, Syngenta, Limagran, Elsams, Ellis Plant Breeding, and of course, BBSRC for funding both projects. Thank you very much.
Thank you to our presenters. I hope you found those talks useful. We'll now continue with information about how to apply and the course scope. While I'm presenting, please do continue to send us your questions using the Q&A function on Zoom. We'll start with a quick update on recent changes to FOF for those of you who are already familiar with previous rounds of the programme. Previously, FOF made both standard and super FOF awards. These have been combined into a single, more flexible award that allows both early and later stage work and which has a continuous funding range from £100,000 up to £800,000. To speed up the time that it takes to process the awards, applications will now go directly to panel review. This means the external peer review stage is removed, and to facilitate this, the size and breadth of the assessment panel has been increased. The requirement for applicants to hold previous BBSRC awards has been expanded to include any direct previous BBSRC funding. We've also updated our guidance on managing intellectual property to be clearer and more flexible. You can find this in the additional information section on the Funding Finder webpage for this round. Applications to FOF will be accepted through the TFS system and will be accepted until 4 p.m. on the 27th of March. The project lead is responsible for completing the application process, and this includes making sure that it's submitted before the deadline. The application can only be submitted by the lead research organisation. To start your proposal, visit the Funding Finder webpage and click the blue button at the top that says Start Application. This will prompt you to log into TFS or to set up an account, after which you'll be able to access the application form and the guidance for how to complete each individual question. To be accepted for assessment, your proposal will need to meet the requirements of the scheme, and this includes eligibility for BBSRC funding. FOF operates under standard UKRI eligibility rules for Research Council grants. This means that you must be employed at an eligible institution at a suitable level or higher, and if you're unsure if you or your institution is eligible, then you can email the eligibility mailbox at BBSRC to check. FOF is intended to enable researchers to progress previously supported work towards impact. As a consequence, there is a requirement for proposals to draw upon current or previous BBSRC investments, and to enable research outputs to be further developed into practical application. Eligible previous BBSRC awards include studentships, fellowships, and work funded by BBSRC through IAAs and other block awards such as centres and networks. If you're unclear, you can email the FOF team mailbox to check. Your award will also need to be within the standard scientific remit of BBSRC. BBSRC funds bioscience with a focus upon plants, microbes, animals, humans, and tools and technologies that underpin biological research. This includes work on all scales, from molecules to landscape. This slide has common queries about our remit. BBSRC does research relating to humans, but does not fund research on specific human diseases or abnormal conditions. In that case, MRC would likely be the relevant funder. To be within BBSRC's remit, the research focus should be upon the biological mechanisms that are relevant to the system or challenge. For proposals with a focus on technology development, we will often ask if the technologies are platforms, rather than clinical or medical interventions. Other areas that commonly lead to questions include animal diseases, where studies primarily focused on animal models of human disease and toxicity are not within BBSRC remit. Research in wild species will tend to be within the remit of NERC, except where they are pests in an agricultural setting or pollinators of agricultural crops, or where they act as a model. For non-terrestrial systems, BBSRC will fund projects related to aquaculture and model species such as zebrafish, but often projects related to marine biology will be supported by NERC. Finally, BBSRC will fund research related to the management of land, but only where this relates to agriculture, horticulture, and aquaculture. Project partners are not a requirement for FOF, however they are encouraged as they strengthen most proposals. Partners can be from business or other user groups. Your partner should add value and be connected to the aims of the project. If your project does not have any partners, then we would advise you to clearly explain how users will be engaged in the development and the outputs of the work. Partners are required to provide a letter of support to confirm that a collaboration agreement will be put in place if the project is successful. Please note that the standard guidance for subcontractors on Research Council grants all applies to follow-on fund awards as well. Moving on from requirements to the actual assessment of the application, each proposal will be assessed in terms of its scientific and technical merit, societal impact, economic impact, and added value. The full descriptions of each of these criteria are provided on the Funding Finder webpage, and this is the same text that we will be providing to the assessment panel to brief them on how to approach the assessment. This slide has an overview of the questions you'll be required to complete when writing your proposal. While I'm not going to read out the text on the web page for each question, the point that I did really want to clarify is that you will find a full description of each question on the web page for the opportunity. This guidance includes a description of what the assessors are looking for, and again, this text is the exact briefing that we will provide to the assessment panel, so you have complete sight of how we will be asking the panel members to approach assessing each question. And please do bear this in mind when writing your responses. So these are the core questions in the set that will form the case for support, and I'd like to talk about these in a little bit more detail. Applicants will be asked to describe the opportunity and the market they are seeking to progress towards, to develop or deploy a new or improved product, process, service, or technology. The application needs to include a development plan that explains how the idea has been developed to date and the proposed approach for further development. When describing route to market and intellectual assets, you should consider how the proposed project will progress the innovation towards market and application. In the section about wider benefits, you can describe the impact beyond the immediate commercial opportunity, including the potential societal, environmental, and economic benefits of the proposed approach. Finally, we ask whether there are any ethical considerations associated with the outputs of the project, including those that are beyond the formal regulatory and legal frameworks that apply. We offer some general advice to applicants based on how proposals have performed in the past. When writing your proposal, we would advise you to take care to ensure that the proposal demonstrates a clear understanding of what the customer or user needs and how those needs will be addressed appropriately in the FOF project. It's important to focus on the most appropriate opportunity for commercialization with a clear development plan that connects to it directly. Any risks should also be addressed directly, with a clear description of any potential impacts and what steps you propose for mitigation. Management of any intellectual property should be clearly described, including how foreground IP will be managed. It's also important to have purposeful and clear project partner contributions or user engagements that directly relate back to the aims of your project. This is the final slide in this section, and it briefly covers the assessment process. After your proposal is accepted, it will proceed directly to expert panel review, provided that it meets all the requirements. FOF proposals do not undergo external peer review, so you will not receive reviewer comments or be required to submit rebuttals. The follow-on fund will recommend directly to BBSRC whether your proposal is suitable for funding based on the published assessment criteria. We expect to inform applicants of the outcome in late July to early August. And as always, your proposal should be written for assessment by experts with relevant but not necessarily specialist expertise. So I'd now like to take the opportunity to briefly highlight some other relevant opportunities for translational work. The first opportunity to mention is BBSRC iCure, which provides dedicated pre-accelerator support for bioscience researchers to validate their business ideas. This is part of the wider iCure program and is open to research and technical staff at any career stage and at any UKRI eligible organisation. You can find out more information by going to www.icureprogram.com. BBSRC also supports the UK Innovation Science and Seed Fund, UK I2S, which provides early stage venture funding for businesses that emerge from the UK's publicly funded science launch base and has proven itself to be a vehicle for startups to get groundbreaking innovations to market. All of these opportunities and more can be found on the UKRI Funding Finder webpage by filtering specifically for BBSRC. We also offer a dedicated newsletter for opportunities relating to innovation that may be of interest to you. As you're developing your proposals, a good way to locate potential partners and resources is the Confer platform, which is supported by the National Centre for Universities and Businesses alongside UKRI. And finally, if you'd like to find out more about the type of awards that BBSRC and other UKRI councils have made, then all publicly available awards can be found on the Gateway to Research portal. I hope that you found the presentation today useful, and thanks again to our guest speakers. We look forward to answering your questions in the Q&A session. Otherwise, do feel free to get in touch with us using the contact details on the screen. Thank you.